So I'm going to carry on from what Stefan's been talking in MPN and give an overview of the current understanding of the molecular pathogenesis of myelodysplastic syndromes, and also this relatively small but important group of overlap syndromes with features of both myeloproliferative disorders and myelodysplastic syndromes. So I'll talk a bit about the different classes of mutations, about complexity, subclonality, and prognosis, and finally about the specificity of some of these mutations for the malignant phenotype. So we have this range of disorders defined by hematological and laboratory criteria. Here are the classical MPNs, here's MDS down here. And although we have these well-recognized classifications, it's very clear from molecular data that these are not discrete entities. We're not going to have a molecular classification that looks anything like this hematological classification. Rather, what we have some just fairly discrete entities, but a spectrum of related disorders. So we're never going to have a clear molecular signature which tells you a patient has CMML, for example. And if we do have a molecular classification, it's going to look very different from this. So at this end, we have excess proliferation, hematopoiesis that's basically effective. At this end, profound dysplasia and ineffective hematopoiesis. So the process of discovery of what's driving these diseases has evolved from cytogenetics, which still remains an important diagnostic test, through to arrays, which played a very big role in understanding the pathogenesis of MPS. ASXL1 was found by a small deletion by array CGH. We've been very interested in these regions of acquired uniparental disomy, which are associated in this example with chromosome 7 and EZH2 mutations. Now we have this avalanche of new data from next-generation sequencing, and we now know of more than 30 or 40 recurrently mutated genes in myeloid disorders. There will be a few more in rare subtypes, and it's possible that mutations are being missed that are outside genes, because it's really the coding sequence, the exome, that's been analyzed to date. But the general feeling is that we now have a pretty complete picture of the pathogenesis of myeloid disorders. But there may be some surprises. I mean, calreticulin was missed by standard exome sequencing for at least a couple of years by the Sanger. So this just groups these acquired abnormalities, and you'll see different versions of this, into the principal classes of things that affect signaling, epigenetic abnormalities, things affecting messenger RNA splicing, transcription factors, repair factors, and cohesins. And we still have this important grouping based on cytogenetics, and these abnormalities here, many of which are prognostically important, don't cleanly map onto what we know about mutations, so we still need to look at these things. Some of these things don't cleanly fit into some of these categories. We don't really know what CEP-BP1 is doing. Calreticulin, as you heard from Professor Constantinescu, probably is going to fit into this signaling box here. And as I was talking about before about classification, there's no abnormality that defines what we classify currently under the WHO as disease entities, apart from BCR-ABL. So BCR-ABL defines CML. There are some very strong associations, so JAK2, MPL, CALR with MPN, CEP-BP1 with atypical CML, mastocytosis, chronic neutrophilic leukemia, and disorders with ring sideroblasts. But these are no precise fit. And finally, I think really because we understand the mechanism better of these classes, particularly signaling proteins, but also epigenetic regulators, these are the principal targets that people are focusing on for new therapeutics. What is happening with these mutations affecting messenger RNA splicing really isn't sufficiently understood for drug targeting to be developed. Similarly, these cohesins, which are probably involved in the long range control of gene expression, it's not clear exactly how they're working. Transcription factors are intrinsically difficult to target. So broadly speaking, these mutations map as follows. So there's a very strong association between a myeloproliferative phenotype and abnormalities of signaling, obviously very clear for the classical MPNs, but also signaling abnormalities are prominent in MDS MPN. Splicing mutations, cohesins, are most prominently mutated in MDS. And epigenetic factors, transcription factors, perhaps even most commonly mutated in MDS-MPN, widely in MDS. And you'll see these categories encroach upon the MPN ellipse here. And these are mainly patients with myelofibrosis or patients who've progressed to myelofibrosis. So just to talk very briefly about signaling abnormalities in MDS-MPN. So about 50% of cases of CMML have activating signaling mutations, but these are much more diverse than in MPN. So you see JAK2 V617F in about 8% of cases, similar frequency in MDS, activating mutations of NRAS, KRAS, and a whole range of other proteins that basically define the process going from growth factor receptors through to RAS signaling. So it's fundamentally growth factor receptor signaling pathways that are deregulated. And there's an association between signaling mutation and so-called proliferative CMML, defined by a white count of 13. So these mutations are more common, but it's far from absolute. But potentially there's a lot of things here that may be targetable. One other signaling abnormality that's worth mentioning is in chronic neutrophilic leukemia, most prominently, also some cases of atypical CML, where a wide range in different reports, but a substantial proportion have activating mutations of the CSF3R gene encoding the GCSF receptor. Two different clusters of mutations, and in the initial publication from Maxon et al. In the New England Journal, there was a suggestion that some of these may be responsive to ruxolitinib. And the reason for that is that the GCSF receptor requires JAK2 for signaling. And trials are underway to see whether that actually stands up. So to move to epigenetic mutations, so epigenetics is defined as heritable, meaning heritable through cell generations, changes in gene expression without any change in DNA sequence. And what this means is basically either methylation of DNA, so methylation of CPG islands, and the relevant proteins for this are DNMT3A, TET2, IDH1/2, and WT1, the Wilms tumor protein. Or modifications of histones, of which there are a very wide number and range, but methylation and acetylation are the most prominent. And the major players here in myeloid malignancies anyway are EZH2 and ASXL1. So this is just taken from a review, and it just illustrates the two extremes of epigenetic regulation. So here you have a gene that's in an off state, the DNA is methylated, there's a particular configuration of histones. These are the histone tails sticking out, and this is a mark which means histone H3, lysine 27, which is trimethylated. And that's a signal or a mark that's associated with absence of transcription. And in this configuration, chromatin is tightly compacted, wound up upon itself. These genes are not transcribed, and they're not available to be transcribed. At the other extreme, genes are on, or they're in a configuration where they can be switched on easily. The DNA is not methylated. We now have a different histone mark. This is lysine 4 trimethylated, which is put on by the MLL family of proteins. We have a different histone configuration, histone H3 subtypes, and the chromatin in this instance is open. So the genes may be transcribed, or they may be transcribable if they're activated by a particular protein binding. So just to put this into context of the mutations that are seen in myeloid disorders, so here you see, this is just a schematic of two nucleosomes. This is DNA winding around here. These are histone tails sticking out. So here's DNA methylation. It's put on by de novo methyltransferases, and DNMT3A is widely mutated across myeloid disorders. I'll put here just the rough figures for MPN, CMML, which is by far the most common overlap disorder, and MDS. That was originally thought to be a very stable mark, but it's now known that it can be removed by hydroxylation, and that's performed by TET2. Very commonly mutated in MDS-MPN, widely in MDS, and also, as Professor Constantinescu said, in a proportion of patients with MPN. I'll go through that quickly. TET2 requires alpha-ketoglutarate, which is provided by isocitrate dehydrogenase 1 or 2, which is also mutated. So there's evidence that there's functional equivalence between IDH mutations and TET2 mutations. 2-hydroxyglutarate, the metabolite that's produced by these mutant enzymes, can also inhibit other enzymes as well. And there are other factors that require alpha-ketoglutarate that affect chromatin. And presented at ASH by, I think it was Ross Levine's group, mutations in Wilms tumor 1, which were really unknown what they did, look like they also have the effect of inhibiting TET2 as well. So a number of different mutations that are all affecting this process of demethylating DNA. The other prominent marks are histones. So this is this mark associated with transcriptional repression, histone H3 lysine 27 trimethyl. That's put on by a complex called polycomb repressive complex 2, of which there are several components, but the catalytic component is EZH2, mutated across the board in myeloid disorders. And probably most importantly of all, because it's mutated, again, in about half cases of CMML, up to a quarter of MDS, is ASXL1, which plays a critical role both downstream and possibly upstream of the PRC2 complex in regulating gene expression by histone marks. So what are all these epigenetic abnormalities doing? So we've got a very clear idea about signaling abnormalities. They're basically turning on growth factor signals when they shouldn't. What epigenetic abnormalities are doing are much more complicated. So we know they're involved in control of gene expression, but what are the key targets, or there are multiple targets, is less clear. One surprise and one clue is that many of these things are mutated in a variety of rare constitutional disorders, developmental disorders. So EZH2, exactly the same inactivating mutations with a disorder called Weaver syndrome, ASXL1, Boehring-Erpitz syndrome. And these are things that don't necessarily have a strong predisposition to hematological malignancies. And there are a lot of other somatically mutated genes that are mutated constitutionally. And one of the prime connections here is, I think, regulation of homeobox genes. So homeobox genes are critical for embryogenesis. They're also critical for hematopoiesis. An emerging function, I'll talk a bit about this towards the end, is providing a stem cell advantage. So what some people are calling these groups of mutations are landscaping mutations. So they may not give rise to an overt malignancy by themselves, but they create a clonal advantage and create an environment. So if you get a mutation like JAK2, it's then much more likely to grow out. So creating a fertile ground for the development of further malignancy. And I think this is a very interesting area of investigation. So to move on about the significance of some of these things, this is a study that was led by Elie Papra-Emmanuel at the Sanger. It's a couple of years old now, but I think it has some very important points. So this was mutation profiling in, I think it was 738 patients with MDS. They're color-coded according to subtype. And what you can see immediately is this huge complexity. A lot of genes are recurrently mutated. And just to put some numbers on here, so there are 13 genes that are mutated at a frequency of more than 3% of cases. There's a further 30 genes in patients at less than 3%. And there are only 4 genes that are mutated in more than 10% of cases. So if you're thinking about designing mutational screening panels, this is a huge complexity to cope with. And you can't ignore this very long tail. So this kind of pattern is seen widely now in malignancy, where you see some genes that are more prominently mutated, but you have this very long tail. So it's estimated that a full third of driver mutations are at a present of less than 5% of cases. So how do you begin to cope with this? Well, one is to try and understand what are the prognostically important factors. What do you really need to focus on? Well, there's multiple studies that have shown that mutations in EZH2, RUNX1, P53, and ASXL1, as well as a complex karyotype, type, are associated with a poor prognosis. Not all these studies have been completely consistent, but it's a pretty consistent picture across the range of myeloid disorders. So one question this study was able to ask was, is the level of mutation important? Because we can see high-level mutations that are clearly part of the main clone. We can pick up lower-level mutations that are clearly subclonal. And this just shows an example for ASXL1 and RUNX1. So this is the great group of patients who are unmutated. These are patients in red who have a clonal mutation, so a high-level mutation. These are patients who are subclonal, so there was another mutation that was clearly at a higher level. And in this study, there's no statistical difference between subclonal and clonal mutations for ASXL1, RUNX1, and a whole load of other genes. So the limit of detection here was round about 5 to 10%, and it's important to pick these mutations up even if they are relatively low level. Whether that's the case for all genes needs to be investigated. There's a paper just out in Leukemia suggesting that IDH mutations may be different. And clearly this needs to be replicated in other studies. And this really needs the assembly of very large international cohorts of thousands of patients to begin to tease apart some of these important prognostic indicators in a form that's really robust. And that's starting to happen. So P53 and complex karyotype are both associated with a very poor prognosis. This was presented at ASH. This is a group of patients that had complex karyotype, and these had P53 mutation here, no P53 mutation here. So you can see very clearly that it is the presence of a P53 mutation that's the critical adverse factor. And it's the association of P53 mutations and complex karyotype, which also makes complex karyotype look like an adverse prognostic factor. One thing across the board is that somatic complexity is bad news. So the greater of number of mutated genes, the worse the prognosis. And this mirrors what we've known for many years in that cytogenetic complexity is usually bad news. So again, this is the Sanger study. This was looking at 111 genes. These are patients with a small number of mutations up here, 3 mutations, 4 mutations, 4 to 5 mutations, greater than 6 mutations. Very clear association with adverse outcome. This is a study led by Eric Solari from Paris looking at CMML. This was only 19 genes. 70% of cases have mutations in 2 or more genes. And again, if we look at cases with 0, 1, 2, 3, or greater than 4 genes, a very strong effect on prognosis. We found similar things with mastocytosis, KIT D816V alone. Patients do relatively well. If you have any other mutation, patients do poorly. And data from Alessandro Vanucci and Ailu Teferi in myelofibrosis, where again, the number of mutated genes, even with a relatively small screen, gives important prognostic information. So how do we factor this all in? We have the technology now for screening large numbers of genes. And on a research basis, that's fantastic, and certainly in the context of clinical trials, that's really necessary. Certainly in the UK, we're really wrestling as to what, on a routine basis, is really important. So cases where you may be deciding whether to transplant or not, that can be useful information, but is this important information for standard MDS patients, normal karyotype AML patients, and critically, who's going to pay? So the uptake in the UK at the moment of this kind of technology is still very slow. Another question is this really the most important indicator? So we can look for mutations, it's technically doable, but is upfront mutation profiling really where we want to go? I'm just going to mention a study that was really led by Jackie Boltwood and Andrea Pelagatti in combination with colleagues from the Sanger. We had a small role in it where they took a series of MDS cases, CD34 positive cells, and did detailed gene expression profiling and built up statistical models to look and try and disentangle all the prognostic elements, focusing on 12 mutated genes and 4 cytogenic alterations, which were the most common to give statistical power. And what you see here is that there's prognostic value, as you'd expect, in everything. So here's genetics, cytogenetics, gene expression, blood counts, basic demographics, all of that lumped together, an IPSS score. But the prognostic power of expression was greater than that of all the others. And when you start doing this in a sort of multivariate analysis, it turns out that the prognostic significance of genetic factors, so mutations, and cytogenetics is entirely captured by gene expression profiling. So prognostic information present in genetics and cytogenetics is mostly contained in expression and basic blood count data. It doesn't add independent prognostic information. So gene expression data provides the greatest prognostic power. Now we know this is difficult to implement in the diagnostic service, particularly with CD34 cells, but this does question in the future as to what is going to be the best approach to evaluate patients. Gene expression profiling may ultimately turn out to be best. And the other point is it's very much a holy grail to be able to profile patients at diagnosis. Obviously, there are very clear associations with some abnormalities and treatments. But minimal residual disease analysis, where it's applicable, remains the most important prognostic factor for individual patients. So gene expression profiling, mutation profiling, and individual monitoring of patients is going to remain to be critical. Finally, just very quickly about the specificity of mutations for the malignant phenotype. Surely you're all aware there have been a series of publications of finding abnormalities in population cohorts of individuals who were not collected for any hematological phenotype. And the most recent iteration of these were these two papers published before Christmas in the New England Journal, looking at very large numbers of cases, either whole exome sequencing or wide screens, where basically you can find individuals in the general population with DNMT3A, ASXL1, TET2 mutations most prominently, without a hematological phenotype. These individuals have a striking increased risk of hematological malignancy, but this sort of hazard ratio does mean that there are a lot of patients with these mutations who have clonal hematopoiesis, much more common in the elderly, who don't necessarily develop a malignancy. And I'll just show you one example which we looked at in detail, a collaboration with Jan Domansky in Sweden, where they were looking at cohorts of elderly individuals from the Uppsala region. They did SNP array profiling. This was a chap here who had a normal SNP array profile at the age of 71. I can't quite read it. Here he acquired this region of UPD on chromosome 4, which slightly diminished, and he's age 91 here, but it's still visible. UPD for chromosome 4 is associated with TET2 mutations, and indeed we found that TET2 mutation a high level in these samples, and it was still present when he was 71. You just can't see it because it's not homozygous. He wasn't investigated in the bone marrow, but he had normal blood counts throughout this period. So basically, for a period of at least 20 years, this person had age-related clonal hematopoiesis driven by a causative TET2 mutation without developing a hematological malignancy. So the clear conclusion from this is that you need to take molecular data in context. You're not going to get a specific diagnosis only from molecular data. So to summarize then, there are more than 40 currently somatically mutated genes identified that can be grouped into 5 principal classes in myeloid disorders. A small subset of genes and overall somatic complexity is prognostically significant, but possibly in the future gene expression profiling, MRD analysis that need to be factored in and will be more powerful. And this cautionary note that driving mutations can be present at a high level in healthy individuals. The finding of a driving mutation is not synonymous with MDS or another disorder. And just very briefly to acknowledge the large number of people we've worked with over the years to accumulate particularly large numbers of patients. Thank you. And I'll hand over now to the third speaker of this session.